All right, so we're moving on to the last portion of the flow of genetic information in cells, and that is translation. That's where we're going to take the instructions in mRNA in the form of ribonucleotides and then translate them to amino acids. So we're going from nucleotide language to amino acid language. Okay, and so in translation, we're going to make polypeptides, or we can just simplify it and say we're going to make proteins using the instructions in mRNA. All right, so we're moving on to the last portion of the flow of genetic information in cells, and that is translation. That's where we're going to take the instructions in mRNA in the form of ribonucleotides and then translate them to amino acids. So we're going from nucleotide language to amino acid language. Okay, and so in translation, we're going to make polypeptides, or we can just simplify it and say we're going to make proteins using the instructions in mRNA. All right, so who are the players in translation? Well, first we have the instructions in the form of messenger RNA. We have our intermediary, our middleman, the transfer RNA, tRNA. And this molecule is going to be involved in bringing in amino acid to the proper place and lining it up with the portion of the mRNA that's encoding that. We then also have the ribosome, and the key feature of the ribosome is the ribosomal RNA. Okay, and the ribosomal RNA, and therefore the ribosome, will be involved in reading the instructions in the mRNA, and the ribosomal RNA will also be involved in catalyzing and forming the polypeptide or the peptide bond between amino acids to make protein. So to recap, we have messenger RNA, the instructions, we have transfer RNA, the gopher, bringing in the amino acid, and the middleman, okay, acting to line up the amino acid with the appropriate portion of the mRNA. And then we have the ribosome with its ribosomal RNA, which is going to be the factory, that is the ribosome. So it'll be the translator, it'll read the message, and it will be the factory. It will make the polypeptide. All right, so who are the players in translation? Well, first we have the instructions in the form of messenger RNA. We have our intermediary, our middleman, the transfer RNA, tRNA. And this molecule is going to be involved in bringing in amino acid to the proper place and lining it up with the portion of the mRNA that's encoding that. We then also have the ribosome, and the key feature of the ribosome is the ribosomal RNA. Okay, and the ribosomal RNA, and therefore the ribosome, will be involved in reading the instructions in the mRNA, and the ribosomal RNA will also be involved in catalyzing and forming the polypeptide or the peptide bond between amino acids to make protein. So to recap, we have messenger RNA, the instructions, we have transfer RNA, the gopher, bringing in the amino acid, and the middleman, okay, acting to line up the amino acid with the appropriate portion of the mRNA. And then we have the ribosome with its ribosomal RNA, which is going to be the factory, that is the ribosome. So it'll be the translator, it'll read the message, and it will be the factory. It will make the polypeptide. Now, about those instructions in mRNA. Remember, we are going from nucleotide language, ribonucleotides in this case, and we're going to translate those into amino acid language, polypeptide, protein. Okay, and so the mRNA, the message, is read as three-letter groups, such as UUU. You can think of these as words. The proper name for these three-letter groups of ribonucleotides, the name is codon or codons. If we look at the table below, what we see is that each codon, each group of three ribonucleotides can be translated as a specific amino acid. Let's take UUU again. Okay, when a ribosome sees UUU, it reads that as phenylalanine, and it knows to put a phenylalanine amino acid at that spot. Another thing to note is that there are start codons. That is where the ribosome knows that it's to start the protein, the polypeptide, at that point. The universal start codon, that is, it's used universally from viruses through prokaryotes through eukaryotes, is AUG. AUG specifies the amino acid methionine. In prokaryotes, just as a fine technical point, the amino acid is a modified methionine called formal methionine. We also have stop codons. Okay, and these would the unequivocal stop codons are UAA and UAG. If you look at those are in pink. If you look at those, you note that there is no amino acid that corresponds to those. That is, when a ribosome lands on a UAA or a UAG codon, there's no incoming tRNA, there's no amino acid, and so in essence, the ribosome sort of shrugs its subunits and says, WTF, what's going on here? I think my business is done here. And it then falls off of the mRNA and goes about its business elsewhere. There is a third stop codon, UGA. This one is a provisional stop codon. It can be used as a stop, or it can encode the amino acid selenocysteine, depending on the needs of the cell. Also important to note is that the genetic code is universal. That is, it's used in this way depicted in this table by viruses, by prokaryotes, and by eukaryotes. Again, we are all related. Another thing to note is that the genetic code is redundant. Let's look at the amino acid leucine. Leucine can be encoded by 4 different codons: CUU, CUC, CUA, and CUG. So regardless of what is in the third place of a codon, whether it's a U, a C, an A, or a G, if we have a CU preceding, then we always get leucine. What this does is it protects us from the effects of mutation. So if there is a mutation that happens at that last position, whether it changes the U to a C or an A or a G, those are still all read as leucine. Okay, let's recap. Translation, the process of going from mRNA, ribonucleotide language, to polypeptide, protein, amino acid language. Okay, so the players, or actually let's go with the process first. So translation, we said, is using the instructions in mRNA to make protein. We've got our transfer RNA, tRNA, which is the middleman. It's going to carry the amino acids to the appropriate place on the mRNA, which will be inside the ribosome. We've got ribosomes, which are the translators as well as the protein factories. We've got our mRNA, which is the plans, the instructions for the protein. We've got amino acids, which are the monomers for the protein. And now, unlike the case with nucleic acids, the monomers, the amino acids, are not high energy molecules. That is, they do not have high energy phosphate bonds, so their energy, such as it is, cannot be used to fuel the formation of the peptide bond. Instead, we need an external source of energy, and that's going to be the ribonucleotide triphosphate, GTP. So instead of ATP, we're going to use GTP as our energy source. All right, let's do this. So we have our mRNA, and we see at the leftmost portion, we see our AUG, our start codon. Now, the two ribosomal subunits, the large subunit and the small subunit, are going to converge on that AUG codon. Incoming to the right of that is the tRNA. Okay, now the tRNA has two ends. It has the upper end, which is where it's going to join with an amino acid, in this case methionine. And when a tRNA has an amino acid attached to it, it's referred to as being charged. The technical term for that is amino acyl tRNA, but I like charged better, so let's go with that. Okay, so we've got the upper end that's going to join up with the methionine to become charged. And then we've got the bottom bit. And you note that has ribonucleotides. It has three of them arranged as what we call an anticodon. What does it mean, anticodon? It means that it's complementary to the codon on the mRNA. That is, the U of the anticodon can interact with the A of the start codon. The A of the anticodon can interact with the U of the start codon. And then finally, the C of the anticodon can interact with the G of the start codon. Okay, so we have the pairing, and this is what's going to allow the tRNA to bring the amino acid, the methionine, to the appropriate place on the mRNA in the ribosome. Okay, now we're cooking. What we've got is we've got the charged tRNA that has the methionine on one end, and it's using its anticodon to align that methionine with the start codon, the AUG. Okay, the ribosome is continuing to proceed down the mRNA, so it's moving from 5' to 3'. So it's going to encounter the second codon, UUA, and incoming on the right, we have our next charged tRNA. This one has a leucine at one end, and then it has its anticodon, AAU, which will allow it to link up with the second codon, UUA. This is so clever. So now we've got the two charged tRNAs, the methionine charged tRNA and the leucine charged tRNA side by side. This puts them in close enough proximity for the ribosomal RNA to form a peptide bond between the two amino acids to form a methionine leucine dipeptide. Okay, this is going to remain tethered to one of the transfer RNAs, okay, and will continue with the ribosome's journey down the mRNA, okay, to continue to decode the information and elongate our incipient polypeptide. Please note that forming a peptide bond requires the energy input of a molecule of GTP. So that is, it's energetically expensive for a cell to make polypeptides. Okay, that is, each peptide bond in a protein, a polypeptide, costs a GTP, which is roughly the equivalent of an ATP. Okay, that's why cells cannot have every metabolic pathway there is. They have the ones that are best fit for their environment. It's like us. We have a certain budget and we use it on things that we're going to use in our environment. We might want to have other things, but those are luxuries, okay, because we're not going to use them, and so they're costing resources that would be taken from other things. This is also why we can use a cell's metabolic capabilities or lack thereof as a way of profiling it, as a way of identifying it. That is, all cells do not have the same metabolic capabilities. So the ribosome is continuing to move along the mRNA. Remember, it's going from 5' to 3'. And so we're coming up on the third codon, GGU, which specifies glycine. Our glycine-charged tRNA enters the ribosome, and we're going to form the next peptide bond between the leucine and the glycine. Notice that the tRNA that originally had the methionine, it is no longer charged. It is moving to the exit site of the ribosome where it will be ejected, okay, and then go back into the cell's pool of tRNAs where it will be charged again with methionine. Also note that incoming is our next charged tRNA. This one has a phenylalanine, so the next codon coming up must be UUU. Remember, UUU encodes phenylalanine. Now we're really cruising. So the ribosome, again, is moving along the mRNA. We've formed a peptide bond between the methionine and the leucine. That were our first two codons and our first two amino acids. We've got the methionine uncharged tRNA being kicked out, being released. We've got the phenylalanine charged tRNA coming in because indeed, as we had as our spoiler for the last slide, our next codon coming up is UUU. So here we see that we are cruising down the mRNA, that is, the ribosome is cruising down the mRNA, and we are translating the codons using tRNAs with their anticodon to match up with the mRNA, and then their amino acid that is charging them. So far, we've encountered AUG for methionine, UUA for leucine, GGU for glycine, and UUU for phenylalanine. So we have a growing polypeptide chain that is still anchored to the ribosome via the tRNA that holds the last amino acid that was added. So we've finally translated all of the codons, and we come to the last coding one, AGA, with our remaining tRNA, which would be for arginine. And after that, we encounter a stop codon, UGA. Note that there is no tRNA that has an anticodon that corresponds to UGA. That is, there is no ACU tRNA anticodon, and so there's no corresponding amino acid that we can put in this place, and so this triggers the polypeptide to be released from the ribosome. And moving on to the next slide, we end up with the new polypeptide being released from the ribosome and now being able to fold properly to assume its tertiary structure. That is the three-dimensional form, which is the active form for the protein. And the ribosome dissociates into its small and large subunits and leaves the mRNA. What happens to the mRNA? Well, it depends. My favorite answer. That is, if this is a stable mRNA, then it's possible that another ribosome is already at the start codon, the first AUG, and is translating it to make more of the same protein. If this is an unstable mRNA, then it's going to go and become degraded. But in any case, we have now finished up with our translation of this particular mRNA. What we have here for this slide is a summary of the translation process. Slightly different, it's from a different text. I think these sort of slides are nice for using to test one's knowledge of what's going on in the process. If you find it useful, use it however works best for you. If not, then discard it.